All right, could I have your attention, please? Can I have your attention, please? We're going to go ahead and get started. Guys, that means quiet down right now, please. Thank you. Very good. So, if you're wondering what's going on right now, if you don't know who I am, Dr. Lin has not gotten a lot shorter and a lot browner. My name is, at least not as far as I know. My name is Dr. Saxena. I'll be teaching the rest of your class. Hopefully you've seen me throughout the semester and or at the beginning of the semester. And so, yeah, I'll be taking you through the end of the semester with this class. So welcome to the second part of cell biology, or I guess I should welcome me since you've all been here. A few points of business. Does this echo a little bit too much, first of all? Let me turn this down. Is that better? Yeah, okay. A couple of points of business before we get started. Austin, do you want to ask your stuff first, or? Yeah. Do you want a mic or?
No. Okay. So, on our website for the announcements, Joe has put up a poll, or a link to a poll you guys can go to. It's a question of how you guys study. Some of the book questions or the short answer questions. I know a lot of you have answered them already, but if you haven't, you can go to on the announcements after class and just vote for whichever one you do most. That would be great. And then another second announcement is the difference between the discussion board and Echo. So I know that some of you lecture capture to watch after class, which is very great. But the asking questions on those PowerPoint lecture capture after, we don't always look at those. The best place to go is to the discussion board. They must have made the sidebar. And they answer questions. We're always checking that and always answering questions on that. And that is also where you get your extra credit point for asking a question from. So if you have questions that you want to answer, from now on, make sure you go to the actual discussion board.
And to reiterate that the lecture capture ability to ask questions is something that your TAs are monitoring during class. So it's a great way if you have a question during class to ask us. But basically for general questions or after hours questions, nobody's checking that. The discussion board is the place to go, right? Yeah.
Right.
Okay, thanks. Anything else? Okay, awesome. All right, so before we get into the biology, I wanted to take us through a couple of things. And because of time constraints this semester, it's going to be the short version, which might be good for you guys, less of me talking about random stuff. But I do want to emphasize some important points. The first thing that you'll hear me say a lot is you have a syllabus, a frequently asked questions document, and something else maybe, but I guess those two at least. Read those really carefully, reread them. It's a good time halfway through the semester to refresh your memory. I don't really entertain questions that have been already answered, and I do that deliberately because I want to focus on the science. So for those of you who are going to email me and say, when's the exam, or how many questions are on it, or what do I need to study, you're not going to get a reply from me. Because on principle, I refuse to do that. You're adults, you're college students, you can read this stuff and learn it and then focus on the science. And because that frees up my time to actually help you with the science, which is what I'm here to teach you. So please go and refresh your memory on how the class works. Here's our grading scale, here's office hours, et cetera, et cetera. So there were no office hours today because, it's the first day of me teaching, but starting Thursday morning, day after tomorrow, these are my office hours. I'll be in the Student Learning Center, so that's 2 floors up from here. If you just go up the stairs out there, and you'll see it right in front of you, it's this big, bright, airy space. If you haven't been yet, it's pretty awesome, newly renovated. And there's a biology section there where your TAs have been, and that's where I'm going to be too. And that's because I don't have an office on East Campus. My office is on West Campus. So instead, I'm going to hold my office hours in the Math and Science Learning Center. So that's going to happen Tuesday, Thursday, 10 to 10:50. In other words, right before class until I have to come down here and teach at 11, right? So, and I'm happy to walk down with folks as we set up our class those days. Except on exam days. So exam days, because we have to prepare, there are no office hours. Or by appointment on West Campus. So I'm very happy to help students with science if you're willing to come see me if you can't make these office hours that are posted here. But please do so in advance. I don't entertain last-minute requests. Believe it or not, professors are not sitting around smoking a pipe waiting for you guys to show up. I need advance notice, and again, that's in your FAQ document, exactly how you should contact and set those appointments up. If you do that, I am thrilled to sit with you and help you understand the material. And on that note, there will be 1 or 2 days, I'll announce it later, that I won't be able to hold this office hour, and I'll be holding extra office hours right before exams as well. So all the information will come to you as needed, and so it's really important you check your emails and stay on top of that. Okay, your TAs. You have some really awesome TAs, 3 of them. These are their office hours. I've been hearing from them that almost no one is showing up. So either you're all doing amazingly well, or you're not taking advantage of the resources that are provided to you. You are strongly encouraged. We set these up on purpose to overlap on every single day almost of the week. So I guess not Mondays, but Wednesdays, Thursdays, Fridays. I'm there Tuesdays and Thursdays. And again, you can make appointments with the TAs as well, again in advance. Yeah. Oh, okay. Oh, that's right. Yes. That's right. So we'll update this, but Joe is going to go to Mondays instead of Thursdays. That way you have somebody available every single day of the week, Monday through Friday. So please take advantage of that. This is the website for this Math and Science Learning Center, 2 floors up, and you can go check out the schedule there as well. Okay. I'm not going to take you through the whole syllabus, obviously, but let me see if there's anything else. Grading, just check this out. I've written it for you. I've given it to you as an equation, and then here's your scale. There are no changes to it. If it sounds like I'm overemphasizing something, it's because every semester this is an issue, right? Students don't, not everyone, of course. And if what I'm saying to you sounds obvious, then just ignore it. You're one of the students who's not going to email me and ask me this. But the information you need, if you're wondering, is right there. Please look at it. So for example, a 74.999 is a C. We're not rounding up. We don't just make up grades. It's what you earn is what you get, all right? Questions, this is how to actually kind of ask questions, and you are, as you've probably noticed, highly encouraged to do that. We are giving you a lot of resources in class, the short questions, the fake homework, the this, the that, office hours. You know, please take advantage of that. And then I encourage questions a lot in class, as Dr. Lynn did, and I'll get to that in a second. Okay, a couple of things to keep, one thing to keep in mind, the last day to withdraw from class is coming up. So you'll have your exam 2 grades by then, and so you'll have enough information to decide what you want to do. Now, the schedule, this is where we are. We are going to focus a lot more on concepts now. Dr. Lin did, in my view, the hard part. He had to get you guys to memorize a bunch of boring stuff that you have to know to understand anything. The analogy I draw is, you know, if you want to swim, you have to learn the strokes. If you want to ride a bike, you have to learn the basics. The basics of most things that you learn are boring. But once you get past that, oftentimes it can be quite interesting. And I think I'm lucky that I get to teach the second half, because now we're going to integrate these ideas that you've heard about that are, you know, just memorizing terms and organelles into concepts. What are cells doing? How are they behaving? And as I like to say, cell biology is genetics in action. So you can tell your genetics professor that his or her class is boring, because genetics is just the DNA. What we do in cell biology is we tell you what the RNA that becomes the protein does. So it's genetics in action is cell biology, and I hope to convey that to you during the second half of class with a lot of videos about how cells are behaving and a lot of concepts about how they interact with each other. That's kind of our goal, and our focus is going to be on vertebrate biology with a slightly clinical flavor, because I know most of you, if not all of you, are interested in careers in medicine, dentistry, pharmacy, graduate school, et cetera, et cetera. So that's kind of how we're going to play out the second half of the class. One thing to be aware of, this exam 3 on April 9th, notice that it's for the lectures from March 12th, that's today, through March 2nd, this one. In other words, this lecture on the 4th is not on exam 3. A couple of reasons for that. Number one, it would only give you a few days to study the material, and when possible, we want to give you more time. So you'll have a solid week from the 2nd to here to study all of the material on this exam, which means just 5 lectures are on it. 1, 2, 3, chapter 16, and then these 2 from chapter 17. So it's not a lot of lectures for that 3rd exam, and that's also deliberate because, in my opinion, this is the hardest part of the class. This is dense stuff I'm going to teach you. It's not easy. You have to focus. You have to practice it. And so I've tried to keep the lectures minimal for that, just 5 lectures for that exam. So then this lecture here, the techniques one, will be then on exam 4. So when you get towards the end of the class and you have to take exam 4, don't forget to study this one here for that exam as well. Okay? Your chapters are indicated here that are relevant in the book to look at as well. All right, a couple of notes. I already mentioned this, but please review the syllabus, the FAQ, these resources that are given to you. Office hours, in my experience, often tends to be staring at imaginary tumbleweeds while I check my email. Please come see me. I mean, if I'm going to be there, if they're going to be there, we would love to talk to you. You don't have to have a question to come to office hours. I've watched some students do well by coming to office hours where other students attend, and just by being there and listening to those other students' questions and the answers, they end up by osmosis picking up more information. Kind of like a second lecture, if you will. So you don't have to have a question to show up. But if you are available, pick one or more of the office hours and come by. The discussion board is growing great, so I have no complaints there. Lecture capture reliability. Every semester I've taught here, so I started teaching here 2.5 years ago. Every semester something goes wrong with lecture capture. You'd think the university would figure it out by now, but they don't seem to. And what that means is that hopefully this is all nicely recorded for you, but there's no guarantee of that. And come to class and encourage your friends to come to class. I will not change the exams because lecture capture failed. Lecture capture is there for you to have a record to study again, not just to look at it for the first time right before an exam. So view it as a resource to help you, not as something to risk relying on. Come to class and take your own notes, and you will find that you'll do okay in this class. All right, ask lots of questions and watch out for flying candy. So one thing I like to do, and I picked this up when I was your age, as we old people like to say, I had an orgo professor, and I hated organic chemistry, and I still hate organic chemistry. Always will. It's just memorization, I tell you. But what I loved about him, the professor, was that he encouraged us to ask questions by throwing candy to us in a big hall like this if we asked a question. Now, I tried that my first semester teaching here, and I almost took somebody's head off with a piece of candy. So I am actually not going to throw candy at you. It's just kind of hard to reach up to the top. I still have the arm strength, but I don't have the accuracy. So the candy is here. And please stop by at the end of class and grab a piece of candy. This is to encourage you to ask questions. If you ask a question of any sort on the material, please come grab some candy, and I'll mix up what I bring to class today. It's Hershey's Kisses. And oddly enough, I find a lot of students, I don't know, maybe people are watching their diets or something, but I don't get a lot of candy takers, but I do get a lot of questions. So that's the more important part anyway, so please do ask. And this means not like, what's the meaning of life, or, yes. Not get out of candy, right? I mean, you can, go ahead. But yeah, so I want you to ask questions of me as I give you the lecture material. And there's a second benefit for you to this. If you may or may not recall from the syllabus, etc., you're only tested on what I teach you. So if you're asking me a bunch of questions and I don't make it through a few slides, those slides are not going to be on your exam, right? So the more you ask, the more you engage, the deeper we go into the material. If you imagine cell biology as a lake, and a lot of people will try to teach you everything on the surface of that lake. I'm not interested in that. I want to dive down into that lake at certain points and have you understand concepts. So if you pause me and ask me good questions and we have a discussion about the material, then you will have less material to learn and you'll understand better what I did teach you. So it's to your benefit, right? You can try to actually, within reason, reduce the amount of material you'll have if you actually ask me questions about what I'm teaching you, all right? So whether you do it for the material, for the candy, please ask about what I'm telling you about. Okay. Normally I give like this long, probably too long thing about my advice for students, and we don't have time for that this semester for a lot of reasons, including the canceled class that we had. But I do want to, I made like my list of the like short list of advice, and maybe some of it's going to be so obvious, and if it is, then great, you're doing fine, ignore it, but I do find it helpful to share my words of pseudo-wisdom, if it can even be called that, with folks. So a couple of things. How do you succeed in this class? That's by far the most common non-scientific question I get. Usually towards the end of the semester where somebody comes up and says, I'm not doing well, I don't know what to change, how do I do better? And it's like, well, it's kind of late, but here are the things you can and should be doing. Okay, attend class and take notes. There's a ton of studies showing that if you come to class and you take your notes, you do better in any subject than if you just watch something on a video. So improve your chances by actually showing up to the class that you paid tuition to come to. And obviously you're here in the room right now, so I'm kind of speaking to the choir, but this is lecture capture, so hopefully your friends who didn't come will hear that, and maybe they'll change their minds and they'll show up too. Okay? Read the book beforehand. Sometimes we get inspired as professors by our best students, and I've had wonderful experiences here. Sometimes we get inspired by our not so great students, and I've had those experiences too. One semester, last week of class, like before the last exam, a student came to my office hours for the first time ever, and the student said to me, only asked one question, and the question was, Professor, should I be reading the book? And I looked at the student and I said, is that supposed to be a joke? And this is where, I'm known, either you're going to like my approach of teaching or you're going to think I'm condescending. It depends on your point of view, right? I don't really tolerate that. And there's nothing I can do to help that student. And that student didn't even ask me any other questions about the material. Once the student understood, yeah, the book should have been read, then that student decided to leave and that was the end of that. Don't do that. Read the book beforehand. You will not be tested on what's in the book. You will only be tested on what's on the lectures. But the lectures build on the book. So if you come in having read chapter 17 or whatever, and then I build upon that, you're going to understand it a lot better than if you're seeing it for the first time. Read the book beforehand. The relevant chapters are right here on your syllabus. All right. Ask questions. We already covered that one, I think, over and over. All right. Do the fake homework, the short questions, all that jazz. We give it to you for a reason. And I suggest finding study partners. You can use a discussion board for that and or the Student Learning Center. It's really helpful to work with people. I'll come back to that in a second. Come to office hours. We talked about that. Practice test-taking skills. This is something I learned as I was teaching here, is that it can be hard sometimes, especially if you're not used to Scantrons or the idea of picking a single choice. And my favorite story about this that always stuck with me was a student who was having trouble with the exam, and we learned that this student was sort of anxious about the test and would read like the first line of the question and then just pick a quick answer and go on. So never having read the whole question, you can't answer the question well, obviously. And so we kind of sat with the student and worked with that individual on, okay, take a breath, read it all the way through. So practice, these A, B, C, D types of questions and eliminating answers. If you get it down to 2 answers, then you know you have a 50-50 chance. What is completely absurd as an answer, could that help me learn about the other answers? So practice those test-taking skills, and that'll help you when you get in the room. We don't want to test your ability to use a Scantron. We want to test your knowledge of cell biology, right? I think, yes?
Actually, a little advice question.
Of course.
I have an issue where if I read the book, I'll get really focused on everything, and I lose all time. So, would you have any? I've asked probably some teachers and still struggling with it. How can I be patient with my studying and not get really obsessed about stuff that you know?
As in, like, you're reading the chapter and you kind of want to pause and go through everything and. That's a hard question. So the question is sort of how do you read, let's say, a chapter without getting too caught up in the details but get through the whole thing? I think, honestly, that I don't have a good answer for you, unfortunately, but I would just say it's kind of everybody's approach is a little bit different. The thing that comes to my mind is when I read scientific papers, I skim very quickly the abstract of the paper, and then I pause and I think about what I just read. So most of the chapters in your book, I think, have headings and maybe even something at the beginning of topics. I would start with those and kind of try to see what those big picture headings are. Sometimes the equivalent that's helpful is if you're looking at a page and reading just the first sentence of each paragraph, and trying to see what that paragraph might be about. And maybe if it helps you, making a list of what you read, like outline yourself. Okay, this is G proteins, whatever. Okay, and then go back and if you have time, read those details. So maybe that would help. I don't know. But your question also, I have something coming up in a second about what kind of a learner you are that I'll come back to, and that might also help a little bit. Teach to learn. This is so important. I use myself as an example. I think students think professors know everything, or hopefully you don't think that. I certainly don't know anything. I remind myself every semester what I'm going to teach you. I'm often looking at the lectures beforehand. I'm going on the internet to remind myself what I'm going to teach you because I have to get it right. And that's the point. When you teach somebody, if I screw up and I tell you the wrong thing, it has a profound effect, right? So I have to get it right. And because I learn to teach, I learn it exactly correctly, and I learn how to communicate that to you. And then that means I really know the material. So why am I telling you this? Be a professor to your friends. If you think you know how G proteins function and how a cycle of G protein activation works, sit with your friend or your colleague and have them ask you, well, how does it work? And then you explain it to them. Draw it out or write it out. And if you find yourself stumbling because you can't remember something, you don't know it yet. So practice that. It's a really wonderful way to learn, is to just teach your colleagues, your friends, your classmates, and then you'll find, or even your family. I mean, make your, I don't know, your boyfriend, your girlfriend, your mom, your dad, somebody sit there and listen to you while you teach the topic to them. It's actually quite helpful. Be a nerd. This is also inspired by a not-so-wonderful former student of mine who went to Austin one semester to complain about me, and this is fantastic. The complaints are fine, I don't mind that. And said to Austin, and I'm going to paraphrase here, you remember this, she's like, I don't understand, what does he expect? Does he want us to be nerds or something? Yes, I do. Think about it this way, you're all wanting to be in sort of healthcare-related or science-related industries. Would you really want to go to your physician or dentist or somebody who just barely skated by and is not a nerd? No, hopefully you want to go to somebody who took the time to learn the material. And so yes, if you're here because you want to do something after you graduate that involves higher education, you need to be a nerd about it. You need to show up. You need to put in the time and effort. Do your part. Do your job. I'm going to do my job to the best of my ability, but if you don't do your job, I can't help you. I won't help you. If you do your part, I will help you a lot. So be a nerd. Just immerse yourself in this material, and I promise you it will pay off for you, because this class is so integral to your careers. This is not, nothing against like English Lit or something, but that's not as integral to, the careers you want. Underwater basket weaving, whatever. This is not one of those classes. You are going to need cell biology in everything you do from now on. So please be a nerd. All right. Now, the last thing then I want to mention as a question for you to think about, and this, I don't know if this helps you with your question. Are you more of a visual or auditory learner? And again, I'll use myself as an example. When I was an undergrad, I hated going to lectures and listening to somebody like me talk. It just wasn't how information sunk in for me, so I would just be in the back sleeping or what have you. And then I realized at some point that reading the material was better for me. I got it from reading whatever it was that I was given because I'm more of a visual learner. I don't like this as much as I do this. You might be different. You might really love a lecture and hate the book. Figure out what you like. Try it. Try different combinations. What happens if you read before you come here? What happens if you read after you come here? What happens if you don't come here and you only read? Figure out what works for you. You're all different. You all have different ways of learning. One is not better than the other. It's really critical for your whole undergrad career and beyond that you figure that out. It'll make your life a lot easier. And your concerns about the book may be something to explore in that way as well. I don't know. Awesome. Right. Right. Okay. So with that ends the kind of blah, blah, blah part of class. Yes? Oh, yeah.
Well, I was just going to say that I feel like the book goes into so much detail. And
Yeah.
I do that I look at the pictures and I read like what it says at the bottom, and if I don't really understand what it still says, then I go and read what the book is saying.
That's a great, I'm glad you mentioned that, thank you. That's a great suggestion for you and other folks, because yeah, I mean, I mentioned like reading the bullet points, but this is even better. I do this too, in fact, and same thing if you're reading papers about research. Going through and looking at the figures in the book and reading the figure legends. It really summarizes what the book is telling you, and that's a great way to start, yeah, absolutely. Good suggestion. Okay, the last non-class related thing I want to share with you is something I've started up, it'll start day after tomorrow, called Cookies and Conversation. And this was prompted by the fact that in academia there's a lot of discussion about the intimidation factor of showing up to office hours or the lack of communication between faculty and students, blah, blah, blah. And so inspired by something somebody else, a colleague of mine has done at a different institution, I recruited 3 other junior faculty, Stephanie Colonia, Alexandra Naba, Yuri Polkanov, and as you can see, we come from different departments, STEM departments. And what we're going to do is basically sit in the Math and Sciences Learning Center on Thursdays from 4:30 to 5:30 p.m., starting this Thursday, and then continuing through April 25th, so right before your semester ends. And we're just going to sit there and have some cookies and have students come by and hopefully talk to us. Hopefully somebody comes by. I mean, if I'm just sitting there eating cookies, that's not good for me or at my age, too many calories, and I'll be bored. So here's what I've written up on this thing. Stop by to have a snack and chat informally with UIC STEM faculty. No agenda, no barriers, just get to know your faculty, and if you like, talk about how to navigate a large research university, your career goals, and other big picture questions. Or just discuss the weather, or the bears, or why everything is hashtag these days, professorial musings, cookies. Okay. Now, what we cannot do is tell you about like degree completion stuff or classes. It's just not, we're not academic advisors, and we don't want to give you the wrong info. But just come talk to us, if you are available at that time. And you can get different perspectives from each of these folks. They're all energetic young faculty who like interacting with students. even me. And so come talk to us. And this is not meant to be like a cell biology office hour, but you know, if you have a question about cell bio and nobody else is there because nobody's eating my cookies, then I can help you with that too. So, okay. With that, now we go to the biology. All right. So now we're going to dive into how cells sense their environment. And before I do that, I'm going to cover a very broad topic, which is pathways, signaling pathways. And I have learned to cover this at the beginning because it tends to trip up a lot of students, so I like to give you the overview and then you have it for the rest of the semester. Okay, there's some terminology that's really critical. Upstream means earlier in a pathway. So in this example here, A, B, and C are random proteins. And what somebody might tell you is that A is upstream of B and C because it's over here. B is upstream of C. What that means is that A is acting on B, B is acting on C. So the terminology is upstream, right? And the word stream is not coincidental. It comes from basically the idea of how a stream would flow, right? So you can think about this as a one-directional flow of information, like water in the rapids here in a waterfall. That's actually a kayaker going down that waterfall, which is pretty badass. But anyway, so you can look at it that way, and the information basically does not go the other way. It's one-directional information. So then if you have this pathway and you say that something is upstream of something else, you say it's earlier in the pathway. So this kayaker is upstream of this pool of water. Downstream, opposite, later in the pathway. B is downstream of A, C is downstream of A and B. This pool of water is downstream of the kayaker. Okay? Basic concepts that you need to know for this class and for many other classes and your careers. Now, this is where it starts to get a little bit complicated. This is just an example of signaling pathways inside of a cell. You know, don't memorize this. This is an example I'm giving you. Here's your cell membrane. Here's your nucleus. And in here, as you can see, are a bunch of arrows and even some upside-down T's that we'll get to in a second. Okay, good. That's big enough to see. In this example, then, you know, A, B, C, this would be an example of that. So A, B, C, D, basically. In other words, RAS is upstream of RAF. RAF is upstream of MEC, or saying it differently, RAF is downstream of RAS and MEC is downstream of RAF. This pathway works in this direction, and starting here, something happens that triggers an effect. It's like dominoes, if you like that better than a river or a stream, and it basically ends up with an effect down here, all right? Now, arrows indicate activation, broadly speaking. So an arrow like this means that MEC is having an activating effect, quote unquote, on ERK. What does that mean? From this diagram, you don't know. So if you imagine that there are 10 molecules of ERK, for example, maybe those 10 molecules are all half asleep or something, and the activation of MEC makes them wide awake. So you didn't change the number of proteins, but you just made them more active. On the other hand, it could also mean that instead of 10, you just made more molecules or more proteins of ERK, and now you've got 20, 50, or whatever. This doesn't tell you which is happening, more of it or more activation of an existing number. Either way, the end result, all this is telling you is that something is changing so that ERK is becoming more active. All right? Upside down T's mean the opposite. They indicate repression. So here's an example of that here. AKT acting on GSK3. Here's an upside down T. What this means is that AKT is inhibiting or repressing the activity of GSK3. So if this was a system and I tossed a bunch of AKT protein into this cell, GSK3 activity would go down. All right? Now, here's where it gets a little bit trickier. This example here, rapamycin, works on mTORC, and mTORC is acting on IRS1. Notice what's happening here. These are both upside down T's, so they're both inhibitions. So mTORC inhibits IRS1, and rapamycin inhibits mTORC. Let's start here and see what happens. Let's say that I have IRS1 protein hanging around, and I add a bunch of mTORC into my system. Is IRS1 activity going to go up or down? Down. Now, what happens if I add rapamycin to the system? Is mTORC going to go up or down? Down. And if mTORC goes down, is IRS1 going to go up or down? Up. That's right. So inhibition of an inhibitor leads to activation. And the analogy for this that I'd like to give folks is math. Oh, I forgot that this is the dam indicating an upside down T, dam in the water flow for the earlier one, this right here. But getting back to the double inhibition, Repression of repression is activation, and the analogy I like to draw to it is mathematics. -1 times -1 equals 1. So in this case, you have a negative, and you have a negative, and the net effect is a positive. So if I on an exam or somebody in your career gives you a pathway that has a bunch of arrows and upside down T's in it, you have to be able to work through and see what happens to the system if you make a change to it. So for example, if, I don't know, let's say I add a bunch of AKT to the system, GSK3 is going to go down, mTORC is going to go up, right, because this is an activation arrow, and if mTORC goes up, then IRS1 is going to go down. If I then inhibit PIP3, then what's the effect on that? You can figure this out once you kind of look at each aspect of it and remember that the arrow is activation, the upside down T is inhibition, and 2 inhibitors will end up resulting in an activation. So play with these. These are good practice slides, just to like make up a scenario for yourself and ask if I up or down regulate the following protein, what happens over here? And as you do that, you'll kind of learn the language. It's like the language of cell signaling, basically. And then the last point on this is that anything written adjacent to an arrow indicates that it acts upon that reaction. So for example here, P10, notice how this arrow is not pointing to any of these proteins or molecules, it's pointing to these arrows. And what that indicates is that here you have a reaction that can run in both directions. Because this arrow is pointing here, it means that it encourages or promotes PIP3 to act upon PI45P2. So, from chemistry, you may remember the term catalyst. Catalysts tend to encourage reactions. SHIP is a catalyst for this activation here. So that's what when you see something written that's pointing not at an actual component, but rather at the arrows, it's making a statement about that reaction. So if this were an upside down T instead of an arrow, it would be telling you that this is inhibiting this progress. All right? So before I move on, any questions about this? I know it can be a little bit complicated. I'm happy to answer any questions. Yes? Sorry, say again, I can't hear you. Correct. That's right, yeah, because the catalyst would be encouraging the reaction, but you can also have inhibitory effects. Yeah, absolutely. Yeah. Any other questions? Okay. All right, so when you can read these types of diagrams, you can learn all sorts of things, such as something you kind of already know through your life, which is that insulin has the opposite effect of adrenaline. You may not realize you knew that, but you actually did know that. Because if you think about what adrenaline does, your fight or flight response, you need energy. So need energy means you need blood glucose, and so one of the things that adrenaline does in your body is it increases the amount of glucose available for your body to use. And as you probably are aware, insulin does the opposite. Insulin decreases your blood glucose and processes it. So whether or not you knew that, this diagram tells you that. Here you're looking at pathways with the activation of basically increasing glucose production, and pathways with insulin decreasing the amount of glucose that's there. And what you might notice is that there's a number of different pathways that converge. One of the fundamental things that keeps us alive and healthy is that we have redundancy. We often have multiple pathways that will do the same end result, so that if one isn't quite working right, the other one can compensate. In this example here, insulin is decreasing your blood glucose by 3 different ways. It's that wrinkling is really loud. Could you keep it down, please? Thank you. All right, so insulin can inhibit the cyclic AMP, and this pathway here, if you follow it through normally with this enzyme, is converting glycogen to glucose. So if you inhibit this pathway, then you're not going to be doing a good job of making glucose from the glycogen. So you've decreased your glucose production by doing this. At the same time, insulin is turning on an enzyme that makes more glycogen from glucose. So now it's also decreasing glucose because you're making less of it here, and you're turning more of it into glycogen. And then third goes around this way and activates a phosphatase that again inhibits this enzyme that normally would have made more glucose. So 3 different ways that insulin is able to inhibit making more glucose and is able to turn the glucose that's already there into glycogen. Redundancy of pathways. Use this as a practice slide to really learn how pathways interact, and it'll help you with the concepts that we're talking about. Okay? And now, with pathways covered, we're going to turn to today's main topic in the time that we have, how cells respond to their environment. Okay. So the few bits of terminology that you have to learn. A ligand. What is a ligand? A ligand is any chemical signal that interacts with and activates a receptor. So it's a very general, broad term. Can be any sort of molecule. This could be a protein, it could be a gas, it could be a chemical of some sort, a sugar, a number of different things. It's a very broad term. Ligands can be diffusible. In other words, they can move through in between cells to get to a different cell. They can be attached to cells or attached to something else. And we're going to cover all of this in a little bit more detail. So here's an example of that in this very amazing diagram. Here's your chemical signal. Here's a receptor on the cell surface. This chemical signal would bind here to this receptor and then start a cascade, that domino effect of signaling the pathways that I just told you about. And those pathways would lead to changes inside of the cell. The receptor then is what binds the ligand. And it changes shape or conformation upon interacting with ligands. This is a pretty common theme about what we'll talk about, and it is true in biology. Most cell signaling happens through shape changes. Proteins or molecules bind to other proteins or molecules, change the shape of the protein, and it tends to change what they do. So it's kind of like, I don't know, maybe my shape when I'm standing still like this is different than my shape when I'm walking. And what you're doing is you're kind of changing that shape to get the protein to do something different. If you imagine this globular protein, it might just alter its angles, and all of a sudden it's able to do something it couldn't do before. Receptors are often integral membrane proteins in the plasma membrane, right here, where they can interact with these ligands that are coming and binding and make something happen. And then yet other receptors are inside the cell, and we'll cover those as well, and can only interact with ligands that somehow get into the cell on their own. So most receptors are here, but there are some inside the cell as well. And the reason, reason that you had sort of evolution lead to having receptors here is that most of these signals are hydrophilic, and your plasma membrane, as you've learned, is hydrophobic, so they can't really get in easily. And so it makes sense for them to bind here and just send the effect into the cell. So in order to do anything, then signals must interact with the receptor. And here again are some examples that we're going to cover of types of signaling. So here's your ligand being released from a cell, and it's binding to this receptor, and it has an effect. You can have other ligands have the same effect. A ligand that has an effect is called an agonist. Now, so an agonist is a type of ligand. Ligand is the broad term. Agonist is one of the subtypes. An agonist has an effect. If that sounds really vague, it's because it is. The effect depends on the context you're talking about. Somebody has to tell you that if you're having a conversation about ligands. Now, in the context of having an agonist that has an effect, an antagonist, the name anti, right, against, opposite. An antagonist does not have an effect. And that antagonist can have no effect two different ways. It can be competitive or non-competitive. The competitive antagonist, as shown here, binds to the binding site that this ligand would have used, and it's basically playing defense. It doesn't let that ligand bind to the receptor. So if you can't bind to the receptor, you can't signal, no effect. A non-competitive antagonist would bind somewhere else, maybe over here, but it's going to change the shape of this receptor so that it can no longer fit the ligand. So imagine in the simplified diagram if this circle-like shape turned into a triangle or something and no longer fit that circle. Now you can't bind and you would have no effect. So competitive and non-competitive antagonists exist. Now I have several definitions for you, including what I just told you. And I'll start off by saying, instead of just trying to memorize these, I encourage you to think about what they mean, because the meaning of these terms is actually within the terms that I'll tell you about. You'll see what I mean. So agonist. Remember I told you an agonist causes an effect, right? There are different types of agonists. They can be full, partial, or inverse. And let's talk about full and partial. If I have a pie, it's a full pie, and if I take a part of it and I eat it, now you have a partial pie, right? That's all that is. A full agonist has high efficacy, high effect. It has a full response through the receptor. And a partial agonist has less of an effect than the full agonist does. That's all it means. And you might be wondering, well, that's vague. How do you know what a full agonist does? You don't. Somebody has to, again, tell you that. Somebody would have to tell you that a full agonist has an effect on a scale of 1 to 10 of, say, 10. And therefore, this other agonist has an effect of, say, 6. So it's partial because 6 is less than 10. It's all context dependent. All right? So you have to know the context. But that's what those terms mean, full or partial agonist. An inverse agonist is perhaps a little bit confusing because it can get confused with an antagonist. Now remember, an antagonist stops the effect from happening, right? An inverse agonist does not do the same thing. An inverse agonist will bind to this receptor, but instead of having no effect, it'll have an effect, but it won't be this effect. It'll be the quote-unquote opposite effect. So an inverse agonist produces an effect opposite to that of an agonist. It binds to the same receptor binding site. So, I don't know, let's say that red is the opposite of blue for the sake of argument. If an agonist made the cell red, then the antagonist would not make it any color, and the inverse agonist would make it blue. All right, so it's having an effect, but it's the opposite or different effect. Okay? Any questions about agonists? Yes? Sorry? An agonist is a type of ligand, or ligand. So yeah, these are all ligands that we're talking about, but agonists are one subcategory that will have an effect. Yeah. Let's see, how are we doing on time? Okay. Allosteric modulators. That's a couple of big words for just a very simple concept. An allosteric modulator binds to the receptor at a site that's not the active site. In other words, not here, but somewhere else. Unfortunately, that chalkboard is covered, so I can't draw this out easily for you. Let's see if my computer will let me draw. Sometimes this works here, and sometimes it doesn't. Okay. So imagine if something bound right here, okay? And then it had an effect. It's not binding to the active site, it's binding somewhere else. And what it's going to do in an allosteric modulator, modulate means to change. So a modulator, an allosteric modulator, is going to change the affinity of the receptor for the ligand. Positive ones increase the affinity, negative ones decrease the affinity. In other words, if I have an allosteric modulator binding here, and it's a positive allosteric modulator, it's going to help this agonist bind better to this receptor and increase the downstream effect. Negative would do the opposite. All right? Now, antagonists, which we already covered briefly, I already mentioned to you, come in competitive and non-competitive forms, each of which that can be reversible or irreversible. This is the easy part. A reversible antagonist simply means that you could wash it off and get rid of the effect. Irreversible means it's bound so tightly you can't get rid of it. Now, again, that's also context dependent. The harder part is this aspect about competitive or non-competitive. Because some of you may have already noticed that when I was describing a non-competitive antagonist to you, I was telling you that it binds over here and it acts as an antagonist, and now I told you about an allosteric modulator, and it sounds like they might be kind of the same thing. They can be the same thing. So this is where these terminologies kind of overlap sometimes. That's just the way it is. So basically, when you have a negative allosteric modulator that is decreasing the affinity of the ligand, let's put this into numbers to make it easier. Let's assume that no effect is 0 and a full effect is 10. And so if you have a competitive antagonist, that's the easy one. It binds here, you get 0 because you have no effect. And if you have an agonist, full agonist, let's pretend your effect is full and you have a 10. All right? Now, if you have a non-competitive antagonist that binds off to the side like this, your effect is going to again be 0 because it's an antagonist, but it's just not binding here. The allosteric modulator that's binding here, my wonderful red squiggly line, doesn't have to have an effect of 0. If it's a negative allosteric modulator, it could bring that 10 down to a 9, an 8, a 5, a 4, or a 0. So to put it another way, a non-competitive antagonist is one type of a negative allosteric modulator. And I highly encourage you to go back and listen to this part of the lecture capture, because I can keep repeating it, but really you have to kind of look at it a couple times and think about what I said, and then it'll all make sense. I promise. Or I hope. So to recap then, agonists make things happen. Antagonists don't have an effect, don't make things happen, and they block the agonist from doing its job. An inverse agonist has an effect, but it's a different effect than the agonist does. Allosteric modulators tweak the effect for better or worse, for up or down. And so that's kind of the gamut that we just covered. And I will make one big picture point here that I'll keep coming back to as well. When we think about changes like this, it's very easy to get into a habit, because this is how we approach stuff in most of our lives, to thinking about it as an effect is good or changes something and an absence of an effect doesn't. So for example, if I turned off that light switch somewhere and this room went dark, obviously lights are better than no lights. There is a positive effect of having lights and a not so great effect of having them off. But when you talk about these signaling pathways, it's not that simple. When you have, let's say, the inhibition of a signaling cascade, that could also have an important effect. In fact, it usually does. Like both, you can think of both states, on and off, as equally important, more like they're forks in a road, where if you go this way with the on state, it's going to do something, and if you go this way with the off state of the signaling pathway, it's not that it doesn't do anything, it's that the absence of that pathway also does something. All right, and I'll come back to that general sort of almost philosophical point throughout the next few lectures, because it's really important to understanding how cell signaling works. It's all a complicated mesh of things being on and off in different ways. So now, we will get into how signals can act over a long or short range. And since we are about 45 minutes in, I'm going to do something I'm going to do each class, which is to take a short break and show you a cool video of signaling in action. Now today's video was very easy to pick because we're covering such a broad topic. Cell signaling, how cells respond to their environment. So I found something that's one of my favorites. No, discard. And let me pause it first here so I can explain what we're going to be looking at. Okay, so this is in a dish. This is a researcher in Australia. She did a really, it's a nice video to watch. This one and this one, these are cancer cells that she put into a dish. And then these are T cells, so lymphocytes, part of your immune system that will go out there and attack cells that aren't doing well and kill them off. What you're going to see encompasses a number of things you're going to learn about in the second half of the semester. You're going to see these cells find these cells, which they do because they're responding to cell signals. Whether or not the cell wants to, it's secreting things that these T cells will detect and say, wait a minute, something's wrong, and they'll go and check these cells out. Then you're going to see the cell bind or kind of grab on here, and at that point it's going to be signaling from inside the T cell into the cell here. And you'll see that that signal tells the cell it's time for you to die. And that's called apoptosis, programmed cell death, something you will also learn about in this class. And then you're going to see the cell turn red, not because it does it naturally, but because this cell has been given some DNA, it's been engineered to express a red fluorescent protein, which you're also going to learn about in this class later. And it expresses that red fluorescent protein when the apoptotic cascade, this programmed cell death pathways are activated. So you'll see the cells turn red and die. And so you'll basically be watching these immune system cells kill these cancer cells because of cell signaling and the downstream effects of it. So let's watch that. It's about 15 seconds long, and it's kind of fun. All right, so there's that T cell, found the cell, and it binds, and boom, cell turns red, and then poof, right? So blebs form. Notice that there's not a bunch of like stuff flying everywhere. Apoptosis, programmed cell death. It's called programmed cell death because it's controlled, it's contained. The body wants to do this, and it does it in a very precise manner. Every day you have thousands, if not millions, of your cells that are dying in this way. Yes? It depends on the context. This video, I don't know the time scale. It's probably a few minutes, right? Just, I'm guessing, maybe up to an hour, a couple of hours. It depends on the context. But this process is pretty rapid. Once you've been told to undergo apoptosis, it can happen in as few as a few minutes. Yeah. So it's pretty cool. You just watch cancer cells die. Imagine if we could just do this all the time. We all get affected by cancer, our friends or family. Imagine if you could find a way to just kill the cancer cells off. Last part of the class, as we get towards the end of the semester, I'll spend a lot of time talking about cancer, which will be of interest to many of you. Yes? So the question was, does it matter the quantity of T cells? That's a good question. In this case, it's just in a dish, I think. So you're looking at 2 cancer cells here, and these are T cells coming in. There's a T cell. There's a T cell coming in. I don't know what these cells are over here. In your body, of course, you will, yes, have a huge invasion. If there's something wrong, it's detected, any T cells in the vicinity that happen to pick up that signal will home to it and attack it. All right, so that's our little, yes?
Cells are eating stem cells so they can create inside of themselves, or is there just the presence of chemicals?
They are, yeah, it's a good question. So they are basically signaling to these cells to activate a pathway for these cells to die.
It's like chemicals.
Yeah, so there are signals on the surface of the T cells that will bind to receptors here, and that will start the cascade. That's called an extrinsic apoptotic pathway. We're going to learn about that in a few weeks. So it's a signaling pathway. Yeah. All right. So back to the not quite as cool to look at, but important lecture slides. Okay. Signals can act over a long or short range. You just saw that. You just saw signals acting over a pretty short range, right? That T cell was not that far from the cancer cell, and then when it was bound up against it, it was acting right up against it. So that's as short range as you can get. So here are 4 examples of, or the 4 major types of cell signaling. And the major difference between them is mostly distance. Endocrine signaling happens over a long distance. The red dots here indicate the signal that's being put out there. The green thing here represents the receptor, and in this case here are the target cells. So this is not to scale, but imagine you've got a cell somewhere in your body, and you've got a cell somewhere else, and it needs to get a hormone to tell this cell to do something. It can often use the bloodstream to send that hormone throughout your body. So for example, if you have a flight or fight response, again with adrenaline, it has to get everywhere quickly. How does it do that? It uses your body's circulatory systems to get around very quickly. So in this case, the hormone is secreted, it gets carried by the bloodstream throughout the body, and where the receptor is present that would bind to that hormone, it activates it. Long distance signaling is endocrine signaling. So sort of the pseudo opposite of that is paracrine signaling. Paracrine signaling happens over short distances. All right, so here's the signaling cell, here's the signaling cell with the red dots that are coming out, and here's the target cell. They're just close by. They're kind of in the vicinity. It spits the signal out, it gets taken up, and you have the response. Then you have synaptic signaling, which is specific to neurons and their axons. So synaptic signaling, here's your cell body of a neuron, here's your long axon, and what happens when neurons work is they'll often build up neurotransmitters at the nerve terminal, at the end of this axon here, and they'll just build up a huge concentration of it until they need it, and it'll spit it out really fast in milliseconds to be taken up by a cell that's directly nearby. Notice that these receptors are super close, but they're not touching. And they'll immediately grab the secreted signal and then have a quick response. So you could make a strong argument that synaptic signaling is a type of paracrine signaling. Because look, these cells are close to each other just like these are. But it is considered a different type of signaling because of the synapse here and the buildup and all that jazz. So you can think of it as a subset of paracrine signaling, but it is its own type of signaling that the synapse of a neuron does, the synaptic signaling. And then the last part is pretty straightforward. When 2 cells touch each other, they can signal directly. In this case, the signal is not being secreted outside the cell. It remains bound to the cell surface of the signaling cell. That's what this line here represents. And it simply is touching the receptor that's on a nearby cell. That T cell that eventually attacked that cancer cell that was bound to it, it was using contact-dependent signaling once it got there to do that. All right? So 4 major types of signaling. There is a 5th type called autocrine signaling. Anybody want to guess what that might be based on the name? Yes, signaling to yourself, auto signaling. If you put out, it may sound a bit odd, but a cell might decide to put a signal out that binds to its own receptor and sends a signal. It does happen. So that's kind of the 5th part, just FYI, or 5th type of signaling. All right. So each cell responds to a limited set of signals depending on its history and its current state. That's a lot of words to encompass. And by the way, these are taken in most cases from your book, but they are summary statements to help you study. I don't give out practice exams, but I do give you these types of things as well as lists beginning and end of lectures to help you study. So when it comes time to study, you should really look at these and make sure you understand each topic. Okay. So each cell responds to a limited set of signals depending on its history and its current state. What does that mean? Well, like I mentioned with, let's say, adrenaline, or in this case with acetylcholine, it's a chemical that signals throughout your body as well. You often want to have different responses to the same signal, and you want to do it quickly and efficiently. So instead of having to send out 10 signals to do 10 different things, what if you could send out the one signal to do 10 different things? How would you do that? How would one signal do 10 different things? It could do 10 different things if different cells had different responses to the same signal. In this case, acetylcholine will have a different response in a heart pacemaker cell than it does in your salivary gland cells in your mouth, than it does in the skeletal muscle cells in your muscles. And in these different cases, it's the same signal binding to a receptor shown here. And notice how this one even is a different receptor than this one. It's actually shown this way to make a point. You could have the same receptor or a different receptor. And then downstream of that, the pathways that we haven't covered yet, those are different. And those different pathways do different things in these cells. So for your heart cell, for example, it changes its rate of firing. Your salivary glands secrete more saliva. Your contraction of your skeletal muscles, these are different effects. So the information conveyed by the signal depends on how the target receives and interprets the signal, which depends on intracellular signaling molecules and effector proteins. These are new terms, right? Intra means within. Cellular means within a cell. Intracellular signaling molecules are signaling molecules inside of a cell. And effector proteins are somewhat poorly named, but are named that way because they have the final effect, whatever that might be. So if you've got a whole signaling pathway and the last one of those proteins makes your cell red or something, that would be your effector protein, the end point of the whole process. And we'll come back to that later. Okay, this is an important general point on this slide. Cells need signals to survive, and that's really important. Imagine if cells inside of me went renegade and started progressing towards an unhealthy state, whether it's cancer or something else. You want to stop that from happening as much as possible. And the way part of how you stop that is by having checks and balances, including the fact that if a cell is not receiving any signals at all because something's gone wrong, let's say it's like, I'm not listening to you other cells, I'm going to do whatever I want to do, it then has, in that situation, when there's no cell signaling coming in, an automatic program for apoptosis that causes it to die. In other words, inside of you, every cell has to be receiving signals from nearby cells to stay alive. It's a good baseline, like checkpoint, safety mechanism, okay? And different signals then tell the cells to do different things, including to survive, to grow and divide sometimes, or maybe to differentiate, and the combination of signals does different things. Any questions about anything so far? All right. A cell's response to a signal can be fast or slow. Pretty simple statement, but let's talk about what that means. So let's say you have your signal binding to your receptor, and now you have stuff happening inside of the cell. Signaling pathways can do a lot of different things. One of the things they can do is take existing proteins. Your cell is full of, a bunch of proteins everywhere, and a bunch of enzymes that are proteins, of course, and other chemicals and molecules. It's a big globby mess. And the fast way to get something to change in a cell is to take those existing proteins that are there and just change what they're doing. Through, for example, the changes in cell shape, the changes in protein shape, conformation changes that I mentioned earlier. If you do that, it can be really fast. If I have a protein that's hanging out and I just tweak it so it then goes and does something else, that can happen really quickly, from less than a second to a few minutes. So, for example, that apoptotic pathway that you saw killing the cancer cells in that video worked quickly enough within minutes because it was altering existing proteins that were there to trigger apoptosis. On the other hand, what if you want to make a profound change to your cell? Sometimes existing proteins aren't enough to do that. You need to really, if I want to turn a stem cell into a neuron or a muscle cell, that is a massive undertaking. It's like instead of moving something in your house from here to here, you want to tear your house down a little bit, renovate it, and bring it up to something entirely different. That requires more than just using the existing proteins, and so you would actually use signaling pathways to change which RNA is being made, which changes what protein's being made, which changes what the cell does. And that can take minutes to hours, or even maybe a day or longer. So you have fast responses that use existing stuff, and slower responses that that make new stuff, basically. And these are not mutually exclusive. So a lot of times a signal will cause an immediate response, but then also set the stage for the longer-term response. So here's some fun videos that encompass some of what I've shown you so far. Let me make sure the sound is on.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
That was Hollywood
Neutrophils
production
are
quality right there. All right, and here's another one, which is kind of fun.
These human neutrophils, taken from the blood of a graduate student, are mobile cells that will quickly migrate to sites of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source. When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
Not sure what that is. So this was in real time, I'm pretty sure. So you can see how quickly that response changes. That's cell signaling, right? A paracrine signaling over a short distance, telling the cell to go here, to go there. And then this last one is pretty fast. This is a cell being held in the pipette, and then this ligand that's being moved around, and you can see again how quickly the cell is responding to this. Actually, it's responding to this endospore that's sending out signals that tell this neutrophil to go to it. So I think this is kind of, yeah, okay, we'll move on. And what about multicellular organisms? Most of you are interested in sort of, well, vertebrates, people, us. Here's zebrafish, a model system that's used in a lot of labs, including my own lab here at UIC. This is not my video, this is somebody else's, but you're going to see lymphocytes moving to, somebody made a wound here in a zebrafish fin, and you're going to see lymphocytes come out from here and move to that wound because they're responding to cell signaling.
To visualize lymphocyte homing to a site of injury, a zebrafish larva was anesthetized and its fin pierced with a needle to introduce a small wound. A vein is seen at the bottom of the frame. Because the fin is very thin and transparent, we can watch directly as lymphocytes crawl out of the blood vessel and migrate towards the wound. They are attracted there by chemicals released from damaged cells, invading bacteria, and other lymphocytes. In a zoomed-out view, we can appreciate that lymphocyte invasion is restricted to the wounded area. The static cells that are dispersed in the connective tissue are fibroblasts. In these movies, 60 minutes of real time are compressed into 15 seconds.
So when you guys go to study this stuff, I think it's helpful to kind of think about these movies because it'll remind you of what the terms are. Oh yeah, there was that zebrafish movie, or there was that cell with the weird sound effects. They might help you kind of remember what the paracrine signaling is, endocrine signaling is, and so on and so forth. And then this is another example, just very quickly from my own work. These are stem cells in green, and this is the nose with neurons in red. And what you're looking at here are stem cells that are going to move towards this area, and then they're going to change their shape and become neurons. They're doing this because they're responding to signals that are being secreted from here that tell those cells where to go and what to do. So, and then here you're actually looking at axons and neurons that are forming and that are going to go end up in the brain. And those axons know where to go because they're responding again to signals that are being sent to them. Okay. So the next thing I want to tell you about is hormones that cross the plasma membrane and bind to intracellular receptors. Remember I told you that most of the time receptors are on the cell surface, but not all the time. Sometimes receptors are actually inside of the cell, and the ligands have to get to the receptors by first entering the cell. Okay, so let's look at that. So this is what I've told you about so far. Cell surface receptor on the plasma membrane. Here's your ligand in red that's binding to it. That's what we've covered thus far. Instead, in this example, your signal is actually hydrophobic. And because it's hydrophobic, it can cross the plasma membrane that most signals cannot cross. So it's able to quickly get inside the cell itself and then bind to a receptor that's inside the cell, often in the nucleus or near the nucleus. And this type of a receptor is called a nuclear receptor or a nuclear hormone receptor. Here's an example of that. Cortisol. Cortisol is a hormone that's actually goes straight into your cell. It'll bind to a nuclear receptor protein. It'll cause a conformational change. Remember, a shape change, like we've been talking about. This has a different shape than this did, and that new shape allows it to basically passage, like as it goes this way, there's a pore here that will let this through because it's in the correct shape to go through, and it goes into the nucleus, binds directly to DNA, and turns on the transcription of certain target genes to make RNA and then protein. So this is an example where a hormone directly becomes part of a protein complex that is able to directly cause changes in the transcription and then eventually, obviously, in translation of particular proteins. Gases can also be ligands, all right? So far we've covered basically solid objects, proteins or chemicals that are binding at the cell surface or that go into the cell. But gases can also cause changes that happen by crossing the plasma membrane. And because they're gases, they can do this very quickly. A very prominent example of that is this is from your book, involves changing your diameter of your blood vessel. So your blood vessel has endothelial cells, and on the outside it has muscle cells that will contract and relax to change the size. This is how you change your blood pressure, and a variety of other related things. Acetylcholine, one of its effects is to bind to endothelial cells, the ones here in dark gray, and inside of those cells stimulate the production of nitric oxide gas, which is produced by nitric oxide synthase. And one thing I forgot to mention, you may already know this, anytime a name ends in ASE, it's an enzyme. I've never found an exception to that. Maybe there is one, I don't know, but if you're stuck on an exam and you see synthase, protease, caspase, assume it's an enzyme if it ends in ASE. So this is an enzyme, NOS, that's going to convert arginine into nitric oxide, and because it's a gas, once that signal is received and that change is made, this gas really quickly leaves the endothelial cells and goes to the directly adjacent smooth muscle cells, causes them to relax through a cascade that we will talk about later, and relaxes the cell. So it's a super fast process. The production of a gas and sending it to a nearby cell to cause a change in that cell. So I keep alluding to this general idea. These receptors are relaying signals via intracellular signaling pathways. What does that really encompass? Personally, I don't like this slide, but I have to teach it to you, and I'll explain why. Both sides of that. What's shown here are intracellular signaling molecules. That's a really vague term. Signaling molecules inside of a cell. That can mean a lot of different things, and it does. And then effector proteins, as I mentioned earlier, are the end result of that. They're also signaling molecules, but they're the ones that are having a direct effect downstream, an enzyme or a protein that does something. But this gets into semantics quite a bit. How do you really decide what's the end result? And why are these called this versus, why isn't everything an intracellular signaling molecule? So I just want you to understand that, because you will have to know these terms, that when people tell you about signaling molecules inside of a cell, intracellular signaling molecules and effector proteins, this is what they're talking about. They're talking about what happens after a signal binds to a receptor and what's going on after that inside of the cell. All right? Looked at it a different way, here's your cell saying, okay, cool, here's what I can do so far that I told you about. I can detect the signal because I have receptors. I can transduce the signal. I can send it inside the cell because I have these proteins that have all these different names that all kind of sort of mean mostly the same thing. Signal transduction proteins, intracellular signaling proteins, second messenger. You may see these, you will see these on your entrance exams, in your careers. They kind of just mean the same thing, with the small exception of effector molecules, which tend to be the last one in that pathway. And that's shown here. I can respond to the signal based on these effector molecules. All right, so that's signal transduction for you. Signal transduction means the communication of a signal from the beginning to the end. That's why these are sometimes called signal transduction proteins. And these are some of the terms from ligand to receptor to this stuff that takes everything into turning it into an actual action inside of a cell. So in the last couple of minutes then, these are the molecules. I already mentioned this, but here it is for you if you'd like to look at it. Groups of these molecules that interact are called intracellular signaling pathways. So when you hear somebody say that, that's what they're talking about. Signaling cascades inside of a cell, same thing. This is just common terminology in the field. It all means the same thing. Cascades, you can think of a waterfall cascading, going back to my analogy of upstream and downstream. It all means the same thing. These are some examples that we'll cover later in the next few lectures. So the last thing I'll end on today, and please don't start packing up or anything because it makes a lot of noise. I will get you out on time, but I want to cover this last thing before we break for today. Positive and negative feedback. Signaling pathways, remember I told you A influences B, influences C, and so on. Sometimes things feed back on themselves. It's like if you give me feedback after the class is over about what you did and didn't like, you're giving me that feedback, right? So positive feedback comes back here from Y to X and tells X, make more of Y. And if you make more of Y in this pathway, then you will make even more of X. This is an amplification. X activates Y or makes more of Y. Y in turn makes more of X or activates X, and it keeps building and building and building. And if there's nothing to stop this, it could go on into perpetuity where you're making more and more of something, of some sort of activity. In reality, of course, signaling pathways don't work that way. There will be a break on this somewhere. But you can think of this as runaway acceleration if it's in isolation. The opposite, in a sense, that this is negative feedback. X activates Y. When Y is activated, it tells X, no, stop doing what you're doing. So the levels or activity of X goes down. As X activity goes down, it's not able to turn on as much of Y. So then Y goes down. And then as Y goes down, the feedback goes down. As the feedback goes down, X is able to come back up. And now you make more Y, and so on and so forth. So if you think about this, here, let's see if I can, I'll draw this real quickly. So let's say X is increasing Y, so Y starts somewhere up here. It's like a high level. And then X gets inhibited, so it's there's not enough X to make Y, so Y starts to come down. And then as that Y comes down, X's inhibition goes down, so then Y can start being made again. Y comes back up, and it can end up doing this kind of a thing. And if there's nothing else going on, ignore that. It's like a bird or something. If nothing else is going on, eventually this can actually stabilize, and you can end up with a steady state where X and Y are in equilibrium. In reality, of course, again, in signaling pathways, you have other inputs coming in that will influence which direction this tends to go in. But that's positive and negative feedback. Positive feedback amplifies the signaling. So in positive feedback, Y would just keep going up, 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 up. And in negative feedback, you have Y going down, but then it can come back up and so on and so forth. With that, we're done for today. We'll end, you're out of here 1 minute early, and we'll pick up on Thursday. Have a nice day. Yes.
Yeah, I have a question. So, as you told, like,